Okay, this is the Vertebrate Zoology Nervous System Lab. We start off by examining the sensory systems and brain structures of the dogfish. So we can begin here by looking at the external sensory systems. So pinned we have the nostrils, an excurrent and an incurrent, the other way around, incurrent and excurrent nostril. And then we have some ampullae of Lorenzini, which are the teeny little dots all over here, which are going to lead to the canals and then to the ampullary organ itself for detection of electroreception. Okay, on the side of a dogfish you can also normally see a lateral line which is visible just here. It's that sort of line of dots, and that of course is for detection of water movement. Okay, so we then move over to our senses internally, and we can start to see the pore and also the duct of the endolymphatic system, which is going to lead to the semicircular canals and ducts, and therefore to the vestibular system overall for the orientation and balance and equilibrium of the body of a shark. We can now see those canals of the ampullae of Lorenzini as well, and we just broadly see other internal structures of the shark. It's worth noting that this is the chondrocranium cartilage, and we'll need to remove that to look at the brain itself. Okay, so if you cut away all that cartilage, you end up with this nice view of our brain, and in this instance we can actually first look at the semicircular canals. So those are visible just here, so there's 1 and 2, and there will be a third one as well. So 3 semicircular canals of the vestibular system. Okay, and then down there will be the sacculus in that little hole. All right, and now I'm going to look at the brain whilst we have an opportunity here, because what we can see is that the rear underneath this cartilage, we would find the medulla oblongata leading to the spinal cord. Medulla oblongata has a large role in homeostasis, but is also the initial sensory system, the initial receiving area for the octavolateralis sensory system, including the lateral line, the vestibular system, and the ampullae of Lorenzini. And you can actually see this superficial ophthalmic nerve, which is going to come back and join to the medulla oblongata underneath this cartilage. We then, moving forward, find the cerebellum, and this is the motor output center, so it's integrating all the information from the octavolateralis system and also vision, scent, etc. to organize motor output by the shark. So really important because it's an active predator. We then have the optic lobes here, which are these 2 bumps, and those 2 there are responsible for visual information. So of course they're connected by the optic nerve to the eye, bringing sensory input integrated, and then we have 3 nerves innervating the eye. The trochlea, the abducens innervate 2 muscles respectively, and then we have the ocular motor, which innervates the other 4 muscles. Okay, we come forward, and now we are at this front region, which of course is the cerebrum. However, it's also called the olfactory lobes in a shark because it's associated with the olfactory centers, and we can see that there is a connection forward via an olfactory tract to the olfactory bulb, which is this region here. So that is actually part of the olfactory bulb there. That should be sticking into the back. There we are. So olfactory bulb, olfactory tract, and olfactory lobes, which of course we better know as the cerebrum. Now, in a shark, like I say, highly specialized for olfactory function, but later vertebrates we see specialization of regions within this for higher level processing of information from across the brain. Okay, so that's our shark, and it's probably worth mentioning that the hindbrain is composed of the medulla oblongata, which is going to be called the myelencephalon, the cerebellum, this is the metencephalon, the midbrain, which is going to be, or the mesencephalon, which is going to be the optic lobes, and then the forebrain, which is actually the telencephalon, which is all of this olfactory structure, and also the diencephalon, which is deep and beneath these structures. And that's going to include the epithalamus, hypothalamus, and thalamus, and the pituitary. So those are all low down underneath. Okay, we carry on. We have the eye, and there's a little bit of eye structure here. The key things to note are really going to be around the function of the different muscles, the optic nerve, and then a pedicle as well. So make sure you're aware that you have a the rectus muscles and the oblique muscles. So for example, you can see here the posterior rectus, and at the front, actually kind of deep down, I'm not sure I can reach it at the moment, there'll be the anterior rectus. And then we have here the dorsal rectus, and this one here is going to be the oblique. Whoops, this one here going sideways is the oblique. Okay. But the key point to note is that those are the muscles, whilst if we were to dig down deeper internally, we would be able to see the optic nerve. I'm trying to find that optic nerve, and there it is. So do you see that white underneath that metal pin number 12? There's a white structure there. That is the optic nerve. Okay, so differentiate that from those muscles. And recall that this is all innervated by the nerve that's there underneath that pin number 12, which is the trochlea. The abducens and the oculomotor are not pinned. Okay. All right. Here we have the cranial nerves, and these are worth noting because your peripheral nervous system is composed of the cranial nerves and the spinal nerves, which integrate with the central nervous system. The cranial nerves, there are 12 of them. And the thing to note is that they have functions that are either sensory, bringing information into the brain, motor, providing information for the senses of the head, movement of the eye, for example, or the facial movements, or they can be sensory and motor, where they are both receiving and sending information in the same nerve. And we can see that, for example, in the vagus nerve, which is number 10, and it's a special one here, X, because the vagus nerve is only one of these nerves that actually leaves the cranium. So it has both a motor and sensory component. It's got an important role in later vertebrates in the parasympathetic nervous system, but in a shark it doesn't, because they don't have an autonomic nervous system, as it were. But that's the only one, number 10, to leave. Other ones to note, because they're visible, I would say would be the superficial ophthalmic nerve, pinned A just here. That one there isn't strictly a cranial nerve, but it serves the ampullary organ in the front of the rostrum. We also should note the optic nerve, which is just here, number 2. So you can see it's pinned into that white structure. And of course that vagus nerve, just here, number 10. And those ones are the highly visible large nerves. Okay. So there's more information on these cards, should it be required. Okay. Right, we're now going to go move over to the spinal cord and the nervous system over here. So we have down these microscopes a suite of images. The first one's just showing some nervous tissue. Okay, we might be able to get in and look at that, but I'm not going to spend time on that actually. Nervous tissue, okay. We're going to have here more examples of a motor neuron. So basically you're going to have neurons which collectively make up nerves. And then we're going to have an image of the spinal cord. It's a cross section, and this one we can probably have a better look at, because I might be able to just bring it in over here. And you can see that it's composed of white and grey matter with a dorsal horn of the grey matter and a ventral horn of the grey matter. Now in later vertebrates, we have a specific dorsal function for the sensory inputs arriving, and then the motor leaving via the ventral horns. And the reason it's all about the grey matter is the dendrites, the nerve cell bodies, and the interneurons. That's where processing really happens. The white matter surrounding that, that's all just myelinated axons. So it's just a connective region. It's of less significance. It's the grey matter where the good stuff happens, as it were. Okay, so worth noting that in earlier vertebrates, prior to the amniotes, the full separation of the sensory and motor between the dorsal versus the ventral had not yet happened. So there's still a component of the motor system in the dorsal in a shark. Okay, we reach our chick embryos now, and the reason these are emphasized is that in development, vertebrates still have a 3-part brain made of the fore, mid, and hind brain, which are also called the prosencephalon, mesencephalon, and the rhombencephalon. And that develops forward towards a 5-part brain. So you need to be able to recognize the 5 regions emerging from the 3 regions as chicks get older. And those 5 regions are the telencephalon, diencephalon coming from the forebrain, the mesencephalon coming from the midbrain, and then we have the cerebellum and the medulla oblongata, which are respectively the metencephalon and the myelencephalon, coming from our hindbrain. Okay, there they all are. Just remember, telencephalon, cerebrum. Diencephalon is the hypothalamus, epithalamus, and thalamus. Mesencephalon is the optic lobes. So just to complement what I said about the others. Okay. We're now going to go along our brain evolution section. So we've got our 5-part brain. What happens? Well, if we begin with our lamprey just here, I'm going to focus on these models because they provide the clearest piece of information. This is a very simple 5-part brain. Lampreys aren't particularly active organisms, so let's think about the lifestyle in relation to these brains. Now, up here we've got olfactory bulbs, cerebrum. Over here's the diencephalon. We've got the optic lobe, the midbrain, and then we have here the medulla oblongata. So that's kind of like the different sections, and you can see this over here. Okay, and there's not really a sort of middle green color here to indicate the cerebellum. It's really not in any visible sense on that model. We go over here to a dogfish, you start to see real changes. Like, look at the size of these olfactory bulbs and the olfactory lobes of the cerebrum. Huge. And that reflects the fact that it's an important sense. Likewise, we have, as we saw in our shark in the dissection, large optic lobes. They would be larger in a shark that's more reliant on vision. So they're smaller in this one. And then we've got a large medulla oblongata for receiving the sensory information from our octavo-lateralis system, and a huge cerebellum for coordinating that information and generating motor output. So it's quite an advanced brain for an active predator. So why are those regions enlarged? Think about that compared to a lamprey. You then compare this to a teleost fish, and you can have a look at this one with its extremely enlarged areas of the optic lobe and a relatively small olfactory bulbs. What does that tell us about a teleost brain, like a carp or a salmon? These are ray-finned fishes. If we compare that to a lamprey or to a shark, you could say, well, clearly it doesn't have a significant olfactory sense for this particular organism, but it does have a really important visual sense. Additionally, and this is important to note, teleosts underwent evolution towards enhanced processing of sensory information and integration in their optic lobe. And that's why this became such a large region, or optic lobes. Together it's called an optic tectum. So that's why that got so big. Enhanced processing in conjunction with it being an important sense. There's a real shark brain here, so make sure you roughly know what that is. Potentially it would be pinned, or we would pin an actual shark so that you could see them in situ. Over here's a Necturus brain, and recall that Necturus are amphibians. It's not a very big brain, and in fact many regions are reduced compared to a shark. For example, the cerebellum is much smaller. Why might that be? Well, are these very, very active predatory animals? They're really not, and they don't even have much in the way of an octavo-lateralis system for the ampullae of Lorenzini in the lateral line. And so as a consequence, you don't have a big cerebellum to coordinate lots of information and generate motor output. So quite a distinct brain. You can see that amphibian brain, which I had put out. Here we are, someone's helpfully moved that. There we go. There's that amphibian brain. And you can see that much reduced cerebellum, large optic lobe, reasonably large cerebrum. And the interesting thing about the amphibians is that they actually start to evolve a hippocampus. This has important consequences because it provides linkage towards the cerebrum from other brain regions, and that is what facilitates the cerebrum rather than the optic lobe becoming the primary processing center. And so we start to see that trajectory going on with alligators all the way through to birds. So that's one evolutionary lineage, right? Expansion of the cerebrum because it starts to do enhanced processing. Birds fly, big cerebellum, okay, and they've got, well, other regions are enclosed, so we can't really see them in this model very easily. Okay, the other lineage, of course, went from amphibians via synapsids to mammals, and we convergently see the same pattern of change towards a large cerebrum with connectivity to the rest of the brain, so lots of processing now occurring in that cerebrum. For example, think about problem solving, emotional regulation and integration, coupled to all the sensory processing. It's now specialized into the cerebrum. And we have a large cerebellum for these relatively active animals. Big expansion in size as well. And an interesting thing to note here is that if we think about a rabbit, it's not got great smarts. Okay, sheep are actually remarkably smart. They can learn to, for example, roll across cattle grids so that they can get out of their fields. They do that, then unfortunately farmers shoot them so that they don't teach the other sheep. Well, not shoot, but they send them to a delightful place where they may or may not get eaten by people. So what's the difference between those two brains? This one is smooth. This one is fissured. And we've got a name for this. The up bits are called the gyri, or the gyrus, and these fissures are called sulcus, or sulci. So that pattern is called gyrification. So we've got enhanced size and gyrification in the sheep brain to increase the surface area. And they do that because, unlike the spinal cord, in the brain you have the white matter on the inside and the grey matter on the outside. So you're enhancing the surface area of grey matter for processing. And it would suggest that probably the cognitive capacities of a sheep are significantly more sophisticated than those of a rabbit. Well, sophisticated is a bit of a judgmental word. Complex would be a better way of saying it. Okay, so we could test that. We should always see if there's a brain region that's enhanced towards a function, we should see greater functionality in that region in the animal in its life. Okay, that's a little bit of a theme to note. So then we have some real brain models for a sheep. So here we are, we can show you that. And here's a nice little cross section. So now you can see in here a cerebellum. Just there, cerebellum. See the medulla oblongata over here. Then we'll also see other regions like the cerebrum all the way over there. Okay, and an interesting note is just down here at the base, there's a region called the pons, and that's really about transmitting information from the cerebellum to the cerebrum and back. And that's because we've got so much of our processing of sensory information now in our cerebrum, we need it to rapidly access our cerebellum to coordinate movement. I'm not going to go into more detail on our mammal brain at this moment. Okay, these are additional brains. I think that's a human. That looks like a cross-section of a human brain to me. Oh yeah, it is. That's kind of fascinating. But this is really just emphasizing that your brain has got all these fissures within it, which are ventricles for the cerebrospinal fluid, and the outside is encased in the meninges, which hold cerebral spinal fluid. So your brain's kind of floating in your skull within the cranium. Okay, so that's this lab. And for reference, here is a shark brain, 5-part brain with the little regions, then the names of those 5 regions, and there are the 3-part developmental brains that those correspond to. Okay.